The overall goal of this procedure is to identify retroviral vector integration sites in the host genome and quantify the relative frequencies of clonal cell populations, each sharing the same integration site.
This method can help answer key questions in retroviral gene therapy, such as which part of the host genome vector has integrated, how genetically engineered cells repopulate after the transplant, and how they are functionally different. The main advantage of this technique is that vector integration site sequences can be analyzed with higher accuracy and sensitivity by sequencing both upstream and downstream vector host DNA junctions simultaneously.
Though this method can provide insight into a safety of vectors and gene-engineered cell in gene therapy settings, it can also be applied to other basic biology studies to investigate individual cell behavior in vivo.
The first step in this procedure is to determine the DNA concentration and the ratio of absorbance at 260 and 280 nanometers of the genomic DNA to be analyzed. After this, dilute 1 to 2 micrograms of the sample genomic DNA to a final volume of 170 microliters of genomic DNA solution using nuclease-free water. Prepare a 200 microliter PCR reaction by mixing the following in a microcentrifuge tube: 2.5 microliters each of HIV-1 specific biotin primers, or a total of 10 microliters for 4 lentivirus specific primers, 20 microliters